So hi and good morning. My name is Christine Lovely. I'm just going to take a step back before I talk to you about my science and just tell you kind of how I got into doing what I do every day. So I grew up on Long Island, not too far from here. And when I was 16, I had this chemistry teacher in high school who said, "Hey, Christine, you really like science. Why don't you go do a summer program at one of our cancer centers here in New York?" So I spent a summer at Roswell Park up in Buffalo, and I loved it. It changed my life in a way that I never looked back from. So I was 16, I worked in a lab, I got to see how science and medicine really come together. That's pretty amazing for somebody who's just in high school to be able to have a glimpse into what that world looks like. I decided when I was 16 that I wanted to be a physician scientist. I wanted to be that person who really understood science on a deep level, but was able to translate it to patients' care to make better treatments. So fast forward 22 years later, when I was 38 years old, I got my first job as a physician scientist, never looked back along that way. And I hope you believe me when I tell you that I love my job every day. It's such an amazing privilege to be able to be involved in patient care and also be involved in developing the therapies that we're actually giving to our patients, moving it seamlessly from the laboratory to the clinic, and then back from the clinic to the laboratory. So I study lung cancer. And as you might be aware, lung cancer is a bad player. So I'm going to fast forward my slides and apologize again that, oops, can you go back one? Sorry. So let's think about lung cancer for a second. Lung cancer is the #1 cause of cancer-related deaths in our country and worldwide. Lung cancer is largely attributed to smoking, but I'm here to tell you that if you breathe, you're at risk for lung cancer. And I'm going to show you one of my patients who was a never smoker who developed lung cancer. This is a huge public health problem and one in which we need to really grapple. There are more than 220,000 cases of lung cancer in the United States every year. If we think about what was the state of the art for lung cancer treatment, this was in the early 2000s. So in the early 2000s, we were treating lung cancer, and most patients with lung cancer are diagnosed at a stage where the cancer has spread throughout their body, a time in which we know that we cannot cure the cancer, but we want to be able to treat it. So this slide shows you what was the state of the art for therapy in lung cancer about 10 years ago, where we were giving all patients chemotherapy. And what you can see on my slide here is that there are 4 different regimens of chemotherapy. The response rate to each of these regimens was 19%. So only 19 out of 100 patients derive benefit. Clearly, we had a long way to go. And again, this is not too long ago. But put this in context of the revolution we're having now with immune therapy and targeted therapy, and you can see how far we've really come. And I think that's why I really show you this slide to put it into context. So where do we go from here? This particular slide shows us if we give chemotherapy in a very non-precise way to any patient with lung cancer, regardless of their tumor genotype or the specific information in their DNA, or regardless of what that patient's particular immune system is telling us, our results are not so good. So how do we make this better? So fast forward now to 2017, where when I see a patient in my clinic with lung cancer, their tumor is actually tested for multiple different mutations within their DNA that help me and other physicians decide how do I best treat this patient's tumor. So we can bring that precision to the clinic. So it's not enough to just say, you have lung cancer, we're going to give you chemotherapy. It's you have lung cancer, you have this mutation in your lung cancer, your immune system looks like this. We are going to develop a treatment plan for you in order to give you the best, most precise therapy that we possibly can. So this pie chart shows you many of the mutations that we test for standardly in patients with lung cancer. But what I want to point out is, unfortunately, there's still a fraction that are unknown. So there's a portion of this pie where a patient comes in and they see me in clinic, and I say to them, I'm sorry, we have not found something in your tumor that will allow me to give you a targeted therapy, a therapy that's specifically directed at your tumor DNA that is often in a pill form. So these targeted therapy medicines are pills, which is actually an incredible advance in and of itself because it allows patients to live the normal lives like we heard from Brent. That is remarkable and cannot be underscored either. So let me tell you about one of my patients. This is Dr. W, and again, I apologize for turning my head to you. Dr. W is a 33-year-old father of 3 children who was a Navy dentist. He was diagnosed very unexpectedly with advanced lung cancer. So let's pause for a second and think about this. Here's a gentleman who just got back from serving our country in Japan, was a father of 3 children, was having a cough. Nobody, most of all him, expected that he was going to get a diagnosis of advanced lung cancer. He had tumor genetic testing of his tumor and actually found this mutation, which was called, it has a complicated name as everything in cancer medicine does, EGFR exon 18 to 25 kinase domain duplication. So there actually are, if you caught it on my last slide, there are EGFR mutations that can occur in lung cancer, but this particular mutation had never been described before. So what did that mean for this patient? It meant 33-year-old, gets a diagnosis of a terminal disease, we sequence his tumor, we don't find anything that anybody knows about right now. And so what the FDA tells us is, unfortunately, we can't give this gentleman the therapies that are already directed at EGFR. So remember, there are EGFR inhibitors in lung cancer. This gentleman has a brand new mutation, never been described before, so we're able to detect it but not do anything about it. That's a horrible feeling to have as a physician and as a scientist, to have something, a finding that I have to tell this gentleman in clinic, I'm sorry, I know you have this mutation in your tumor, but I can't do anything about it. And I know there's a drug that can potentially help you, but I can't give it to you because there's no evidence to support this new mutation with this existing drug. This is not good enough either. This is where the science really comes into play, where we have to be able to study patients' tumors individually and build up the evidence that we need. So because he was ineligible for targeted therapy, again, because this mutation had never been reported before, he was treated with standard chemotherapy. That chemotherapy that I showed you on the previous slide, where only 19% of patients respond. This is certainly not what we were hoping for. While he was receiving chemotherapy, we actually took his tumor back to the lab and started studying it. So we wanted to be able to say, you know what, we have to give this gentleman, this father of 3 children, the best therapy we can. We have to understand what his tumor is telling us, because that is going to help us to give him better therapy. So he got regular chemotherapy. Unfortunately, his disease progressed, and our question was, what do we do next for Dr. W? How do we better deliver therapy to him? So like I said, we took his tumor back to the lab and we asked several questions. First, we wanted to know, is he the only patient who has this new mutation in his tumor? Probably not, but no one had ever looked for it before. So my lab worked with several other groups at Foundation Medicine, at Memorial Sloan Kettering, looking at the Cancer Genome Atlas, and actually found that this mutation is found in numerous other types of cancer. So other patients with lung cancer, some patients with brain cancer, some patients with soft tissue tumors. So this is not an N of 1 experience. This is an example of, if we study it now, we can help the next patient who is found to have this same mutation. We went to our colleagues who are experts in structure and protein and said, help us to understand, we have this mutation in a DNA, but DNA ultimately becomes a protein, and the protein is what is enacting the function in the tumor cells that's causing all the nasty effects that we think of for cancer. We said, help us to build a computational model of what we think this new mutation looks like on a protein level, so that we can understand how it functions, why it's driving his tumor growth, and how we can target it better. Then we actually made a cell line from the patient's tumor himself. So we took his tumor sample, grew it in the laboratory, and tested numerous drugs against it, and found that, lo and behold, there was a targeted therapy called afatinib, which is actually quite potent in inhibiting the growth of his tumor cells. And that's what I'm showing you on the left-hand panel. So meanwhile, my patient is getting chemotherapy. We're building up this evidence in the laboratory. We're trying to understand, how does this new mutation make his tumor grow, and how can we target it? So that when he is now ready for his second line of therapy, we're able to, based on the laboratory studies, give him one of these targeted therapies, give him a pill therapy that is precise, that is directed specifically at his tumor, so that we can actually say that there's rationale for what we're doing. And I'm happy to say that he had a beautiful response. So this is his CT scans, and I apologize for not having a laser pointer, but how I describe these to my patients is, The top is the front of the patient, and the back is the back of the patient, and the white spot where the arrow, behind the arrow is his spine, and the red arrow is pointed to his tumor. So he had more than a 50% shrinkage in his tumor by understanding, here's the mutation in your tumor, we're going to build laboratory-based evidence, we're going to show that this mutation can be inhibited by a targeted therapy, we're going to deliver that pill to you, and by the way, this pill was existing. We just could not give it to the patient because there was not the critical evidence needed to be able to say to regulatory authorities, here's a rationale for using this drug in this patient. So this is a great example of repurposing drugs that are existing already based on really good science and good evidence. So what is the impact of this? So we found in this one case, one mutation, and there are thousands that can occur in cancer, found that this mutation was recurrent to other patients. If we understand this better, there are other patients who would benefit from these therapies. We found that there was not just lung cancer patients who had this mutation, but other types of cancer as well. And really where this is going is, and a big push in my laboratory is to, as we have very, very sophisticated sequencing of our tumor samples now, we have to build that critical evidence to say, we found a mutation, now we have to do something about it. Because it's not enough to just find the mutation, to do the sequencing. We have to be able to say, give us the scientific evidence to take that mutation to a therapy, to build precision into the patient care that we give for all of our patients. So one last thing that I've been very fortunate to be involved with on my 3.5 years on faculty now is developing this website called My Cancer Genome. And the reason I show you this is because the advancements that we are privileged to be a part of in cancer medicine occur so fast and at such a rapid pace that it's hard for even me. I live, eat, and breathe this. I love what I do, and I can't keep up with it. So how do we help physicians in community practice to be able to keep up with the same kind of advances? Because I don't care whether you're in New York City at Memorial Sloan Kettering, or you're in Nashville at Vanderbilt University, or you're in rural Alaska, if you are a cancer patient, you are entitled to the best therapy that anybody can give anywhere in the country and anywhere in the world. And it is our job as a community of oncologists to be able to educate each other and say, here's what this mutation means, here's what this T-cell therapy means, here's how you give the best therapy to your patients wherever you're seeing them. So the goal of this website is really to provide physicians at the point of care. So when they're seeing a patient, if you find a mutation on your patient's report and you don't know what it is, you can go to the website, and this is one of many now, where you can look up the information and get evidence of what do you do with this mutation once you found it. So you can go to your patient and say, you have this mutation in your tumor, here's what the most rigorous scientific and clinical trials are telling us for how we should treat you. So last but not least, I absolutely cannot end my conversation without showing you some important pictures. So this is my team. This is my generation of scientists that are my babies in the lab. And I just graduated my first PhD student. So that to me is such a huge benchmark to say that a young scientist now came out of my laboratory and has a PhD as a result. And it is unbelievably humbling for me, still as a young scientist, to be here amongst people like Dr. Kalin and Dr. Thompson and Dr. Cantley, who are the heroes in my scientific world, who are the people that I always looked up to when I was a graduate student. And so I hope that I can bring my love of what I do to everybody in my lab as well. And then certainly not last but not least, I wanted to introduce you to the patients that have really touched me. So on my right-hand side is Dr. W, whose name is Ned, who unfortunately passed away about 6 months ago. And I'm sorry, and it's still very sad about it. We need to do better, because it is not a happy day when you're an oncologist and your patient passes away. So I show you Ned. He was very involved in our research. He came to the lab. He looked at the cells that were growing in tumor in the incubators in my laboratory, and he was just excited to be part of the research and excited to know that what he was going through would help the next patient who has the same kind of mutation in his tumor. And on the left-hand side is Nicole. She was an 18-year-old who was diagnosed with high-grade sarcoma, who had something called ALK in her tumor, another target that I study in my lab. Unfortunately, she passed away too in January. And I show you them to not only commemorate their amazing spirits, And here we're celebrating all the research that we have, but we still have a long way to go. And we want to be able to help people and our young patients and all of our patients to be able to give them the best therapies we can. Thank you so much for your time.